In this chapter, we discuss the regulation of eating behavior and body weight. Be able to recognize terminology and be familiar with the learning objectives that are posted in Moodle for this chapter. First topic of discussion is homeostasis. So we need to define homeostasis. It is defined as the stability of the body's internal environment, or the ability to maintain balance of the internal environment in the body. There's a theory that the regulation of how much fat we store may involve a set point or a reference value. If we do, in fact, have a set point weight around which our body wants to be maintained, it could defeat or make very difficult our attempts to reduce our fat stores. In the maintenance of homeostasis, there is a graphic in your textbook in this chapter. It shows that receptors detect a stimulus first and then sends that to what's called an integrator. Integrator gets a signal. If this is body weight regulation, the receptors are nutrient sensors that are located primarily in our brain, gastrointestinal tract, and additionally within our fat tissue. The job of the integrator is to collect information from receptors and to coordinate a response. The effector's job is to carry out the response. And if this is body weight regulation, effectors may produce endocrine hormones, which can increase or decrease the activity of body tissue. And the response is a change in energy in and energy out. The brain is a central regulator of our weight. Most of our internal mechanisms turn on our appetite or turn on eating. Hunger, be familiar with these definitions, is defined as a drive to obtain food that leads to discomfort, physical discomfort such as hunger pains. Appetite, on the other hand, is defined as the psychological desire to eat. Whether there is hunger or not. For example, your appetite can be stimulated by a commercial, but if you've not eaten for 12 hours, you are likely to be hungry. You may have an appetite for something, but hunger is the physiological state. Satiation and satiety. Satiation refers to the termination of eating after hunger is satisfied, while satiety refers to the period of feeling full or feeling satisfied from eating. The graphic here shows the relationship of the peripheral nervous system, autonomic nerves, somatic nerves, parasympathetic, and sympathetic nerves to the central nervous system. The most significant area of the brain responsible for regulation of eating behavior is the hypothalamus. The hypothalamus includes 5 different areas, which we'll talk about. The autonomic nervous system is the branch of nerves that regulates physiological functions that are outside of our voluntary control, like circulation of blood, digestion of food, sweating. The endocrine system includes glands as well as hormones that secrete and deliver messages to target cells throughout the body. Part of the pancreas is an endocrine organ. There are 5 areas of the hypothalamus that are involved in eating behavior. They're listed here. They include the arcuate nucleus, the paraventricular nucleus, the ventromedial hypothalamic nucleus, the dorsal medial hypothalamic nucleus, and the lateral hypothalamic area. The ARC, short for arcuate nucleus, is a hub. It contains both neurons that produce appetite-stimulating neurotransmitters and neurons that produce appetite-dampening neurotransmitters. This area also contains many insulin and leptin receptors. The PVN, short for paraventricular nucleus. There are appetite-stimulating neurons that project from the ARC into the PVN. The ventromedial hypothalamic nucleus is an area that promotes fullness or satiety and is very sensitive to the hormone leptin. We'll talk about leptin. Leptin is a hormone that plays a very significant role in the regulation of eating behavior. If the VMH is damaged, it will result in uncontrolled eating. It may result in excessive weight gain and may result in a reduction in the sympathetic nervous system activity. Dorsal medial hypothalamic nucleus, or the DMN, is not very well understood. It may act as an integrator. The LHA, or lateral hypothalamic area, this is a feeding center. It is very sensitive to hypoglycemia. This area contains neurons that produce neurotransmitters called orexins. Orexins stimulate appetite. The destruction of the lateral area will result in failure to eat and extreme weight loss. This graphic is from the internet and it shows the limbic system and its parts. The hypothalamus is part of the limbic system. The next topic of discussion focuses on neurotransmitters that turn on feeding or turn on our desire to eat. They include NPY, or neuropeptide Y. This is actually the most abundant neuropeptide or neurotransmitter in our body. It's found in the central nervous system. And it seems to drive the consumption of high carbohydrate foods. NPY is produced by the arcuate hypothalamic nucleus, ARC. When food is consumed, levels of NPY decrease. NPY can decrease energy expenditure by reducing or inhibiting the actions of the sympathetic nervous system in brown adipose tissue. Brown adipose tissue is a good type of fat tissue to have, and we'll talk more about it. Agouti gene related protein known as AGRP. It is associated with the consumption of high fat foods. As food is consumed, levels of AGRP fall, just like with neuropeptide Y. Melanin concentrating hormone, or MCH, is also produced by the ARC and the LHA, or the lateral hypothalamic area. In laboratory mice that were studied who had low melanin concentrating hormone, they became very lean, whereas laboratory mice who had too much melanin concentrating hormone overate, particularly overate high-fat foods, and they became obese. Okay, so excess melanin concentrating hormones was associated with obesity in mice. Other neurotransmitters that turn on feeding include orexins, ghrelin, and dynorphin. Orexins are produced in the lateral hypothalamic area, or LHA. They are neurotransmitters that stimulate eating. They also play a role in sleep regulation. Ghrelin. Ghrelin is a neurotransmitter that is associated with an increase in fat consumption in the diet and an increase in body weight. Levels of ghrelin increase once we hit puberty. If you are food restricted or you're fasting for some reason, levels decrease. It's interesting. NPY is another neurotransmitter that increases during times of food restriction. Dynorphin is what's called an opioid peptide. Opioid peptides tend to have pain-reducing properties. It promotes eating and may play a role in stress-related eating. If you get stressed out and you eat, dynorphin may be playing a role. It promotes eating and pain reduction. All right, let's talk about norepinephrine and alpha-melanocyte stimulating hormone. Norepinephrine is a neurotransmitter that acts on the part of the hypothalamus known as the PVN, or paraventricular nucleus. The paraventricular nucleus may play a role in stimulating feeding. Norepinephrine drives one to increase their meal size and increase the amount of carbohydrates you intake, or carbohydrate cravings. If you overeat, in response, norepinephrine is going to increase in your brown adipose tissue and induce thermogenesis. If you undereat or you're food restricted, the opposite will occur and result in reduced energy expenditure, possibly weight gain. Food restriction decreases release in brown adipose tissue. The next section introduces you to neurotransmitters that turn off feeding. The hypothalamus produces catecholamines that can turn off feeding and in some cases can increase energy expenditure. Catecholamines tend to promote fat loss. They are known as anorexigenic peptides, and neurotransmitters include those listed here and in your textbook, specifically pro-opiomelanocortin, also known as POMC, alpha melanocyte stimulating hormone, cocaine and amphetamine regulated transcript, serotonin, cholecystokinin, also known as CCK, peptide YY, and amylin. All right, pro-opiomelanocortin derivatives are produced in the ARC, or arcuate hypothalamic nucleus. They oppose the action of melanin concentrating hormone. Melanin concentrating hormone stimulates eating, okay, and POMC derivatives oppose melanin concentrating hormone. Serotonin is a catecholamine that opposes norepinephrine. When serotonin levels are high in the brain, the rate of eating slows, the quantity of food consumed decreases, and the duration of eating are all reduced. When we get to the section on drugs and pharmaceuticals, we will discuss serotonin. There's some antidepressants that can increase the amount of serotonin circulating in your body and therefore can induce decreased appetite in some people. This chart is from your textbook and shows peptides that are responsible for the regulation of food intake. There are a number of them. The peptides that regulate food intake in terms of turning on feeding are given the name orexigenic peptides, while those that turn off feeding are referred to as anorexigenic peptides. The peptides that have an asterisk by them in this chart indicate ones that are produced in or act on the digestive system. There are some mechanisms called non-homeostatic mechanisms that influence energy balance. These are the unconscious actions of the hypothalamus. They can override hunger signals. There's a group of brain structures called the circumventricular organs. They're located around the ventricles of the brain, specifically the third and fourth ventricles. They're outside the blood-brain barrier, and therefore changes in the amount of circulating hormones or peptides or glucose can cause a very immediate response. The limbic system, which was pictured in a previous slide, is a ring of interconnected structures that functions in pleasure and reward, the dopamine reward system. It plays a role in motivation, emotions, learning. Insulin may decrease pleasurable response to food. The key areas of the brain are shown here. Take note of the third and fourth ventricles. This is the area of the circumventricular organs. The limbic system, again, is a ring of interconnected structures in the brain. It includes part of the hypothalamus, the thalamus, and cerebral cortex, and plays a role in motivation, learning, emotion, reward, pleasure. The digestive system carries out eating responses directed by the brain. In response to a stimulus, such as your nutritional state or the taste and smell of food or food memory, receptors receive signals Receptors are located in the digestive system, our adipose tissue, and in our peripheral nervous system. A response and integrator is stimulated. Integrators include the hypothalamus and other brain areas. This in turn stimulates release of effectors. Effectors include neurochemicals, hormones, metabolites, and adipokines that stimulate a response, which will change your energy expenditure and or your food intake. Okay, let us look at the digestive system and its role in the regulation of eating behavior. If you ever had a cold and lost your ability to smell, you probably lost your appetite as well. The tongue plays a role in eating, as does the small intestine. Our taste receptors, our smell receptors, play a role in our appetite, and they're responsible for sensory specific fullness or satiety. Recall that there's a psychological basis for intake and appetite. Our stomachs have the ability to shrink and stretch. An empty stomach holds about 50 milliliters. That's about a fourth of a cup. Fluid, if you're looking at fluid, about a fourth of a cup, or a couple ounces. And as food is consumed, the stomach's smooth wall muscles stretch and relax simultaneously to accommodate an increase in volume to hold up to 1 liter. This is called stomach distension. When the stomach distends, it will promote stomach emptying into the duodenum, which is the upper part of the small intestine, and stomach distension will also signal satiety or fullness by activating stretch receptors. Signals get sent to the brain that result in a slower rate of eating. A high fiber diet can promote fullness or satiety by absorbing and holding more water in the stomach. Okay, so try to get at least 25 grams of fiber in your diet every day. If you're getting 25 grams of fiber a day in your diet, you're winning. You're already ahead. Okay. If you're not, try to get there and do so gradually. I cannot stress the importance of fiber enough. I really can't. Okay, hunger and digestion. There's only one hormone in the GI tract that stimulates food intake, and that is ghrelin. It is secreted primarily in the stomach. It reaches peak levels right before you eat. Ghrelin activates our limbic system. There has been some speculation that ghrelin may play a role in helping people find food. Ghrelin levels will increase after you fast, so this is your body trying to stimulate the limbic system to increase food intake. Your body doesn't really like to fast. Or levels of ghrelin will increase after calorie restriction. Satiety in the digestive system is the topic and function of the neuropeptides listed on this slide. Satiety or fullness is stimulated by the hormone cholecystokinin or CCK. It inhibits neuropeptide Y to slow down eating. Some studies have showed that there's decreased number of CCK receptors in obese rats, and there may be decreased CCK receptors in some obese. Peptide YY is responsible for inhibiting NPY as well. It is produced in the small intestine after eating. Bombasin is secreted by the stomach, and glucagon and amylin are secreted by the pancreas. Glucagon and amylin are responsible for producing a short-term feeling of fullness or satiety. These peptides can send a satiety signal to the brain within minutes. Insulin plays a role in hunger and satiety. Insulin is secreted by the pancreas, and the hormone glucagon is also secreted by the pancreas. We have insulin receptors in our hypothalamus. When glucose levels are high and insulin is high in the ventromedial hypothalamic nucleus, this will slow eating. If high insulin levels are associated with low blood glucose, or you have low blood glucose with high insulin levels, this is called hyperinsulinemia. You have too much insulin in your blood. This can be the case in a lot of people who have type 2 diabetes, and having excess insulin or hyperinsulinemia predisposes you to nerve damage. When insulin levels are low and blood glucose levels are low, it will stimulate NPY, which will stimulate eating. After you lose weight, insulin may play a role in restoring your set point weight through calorie restriction. All right, so make sure you read about insulin, hunger, and satiety in your textbook. I know that's a lot of information. This is an optional application in your textbook. If you have the 3rd edition, it is in chapter 5. If you have the 2nd edition, it is in chapter 4. This application introduces the Rise family. They have some problems with obesity and some chronic disease, and I encourage you to do this optional application as practice. I might ask you to turn in one of these practice applications that I've encouraged you to do during the semester. I might ask you to turn that in to me for a bonus at the end of the semester if you need points. So go ahead and use this as practice for pulling all the information together. There are 3 parts to this application. Stored fat is a term that is used synonymously with white adipose tissue or simply adipose tissue. Stored fat can act as a receptor and an effector. It can detect its own size, thus acting as a receptor. And as an effector, can actually respond to both hormonal and neuronal signals, or signals that come from your nervous system. Fat cell development is mentioned in your textbook, and there is a chart that shows the hyperplasia and hypertrophy of fat cells. Storage fat can act as an endocrine organ and may play a role in body inflammation. During the second trimester, white adipose tissue development is underway. It continues after birth. The average non-obese individual, this comes from your textbook, has between 30 and 50 billion adipocytes. The enlargement of fat cells is referred to as hypertrophy. Hypertrophy is reversible, while the increase in number of fat cells refers to hyperplasia, which is not so reversible unless you have a surgical procedure to remove that fat or liposuction. Adipocyte formation is talked about in your text. Pluripotential stem cells become committed stem cells. These committed stem cells turn into adipoblasts, which turn into pre-adipocytes, which turn into adipocytes. The hormone cortisol, which plays a role in stress in the body and inflammation, is responsible in part for the stimulation of pre-adipocytes into adipocytes, as well as insulin plays a role. This is from your textbook, and it shows the structure of an adipocyte and adipose tissues. It looks like a honeycomb. The next section expands on adipocyte growth. The average non-obese person has between 30 and 50 billion adipocytes. There's approximately 0.4 to 0.5 micrograms of fat per adipocyte cell, while people who are obese tend to have more micrograms of fat per cell on average compared to non-obese people. Hypertrophy refers to the enlargement of fat cells in this chapter, and hypertrophy is reversible. When your fat cells hypertrophy to 0.8 micrograms per cell, this may be a point in which your fat cells stimulate the filling of pre-adipocytes with fat to become adipocytes. Hyperplasia refers to an increase in the fat cell number. This is more permanent. Less surgery or liposuction is done. Fat cell development theory is illustrated here. During growth, fat cells increase in number. So while you're growing up until you reach your early 20s, your fat cells will grow in number. There will be hyperplasia of fat cells. When energy intake exceeds energy expenditure, fat cells will increase in size once you are an adult. So they will undergo hypertrophy. When fat cells have reached their maximum size and energy intake continues to exceed energy expenditure, we have hyperplasia again, where we have the additional pre-adipocytes being filled. With fat loss, the size of the fat cells will shrink, but the number of fat cells are not. So hyperplasia is permanent, hypertrophy of fat cells is not. Be able to answer the question, why excess pre-adipocytes fill and become adipocytes? Hyperplasia is said to occur at least into early adulthood during our prenatal period, our early childhood period, and adolescent period. whereas hypertrophy occurs once we're adults, once we have a set number of fat cells. If conditions are right, however, adults can and do experience hyperplasia of fat cells. Know the difference between lipogenesis and lipolysis and how they're regulated. Lipogenesis refers to the accumulation or production of fat, which are taken up by fatty acids into adipocytes. It is the creation of new adipocytes. It is regulated by lipoprotein lipase enzyme, acylation stimulating protein, and insulin. Lipolysis refers to fat breakdown. Fat breakdown results in a release of fatty acids into the bloodstream. It is regulated by hormone-sensitive lipase. Lipase enzymes break down fat. Fat cell number is determined by the balance between lipogenesis and lipolysis. The autonomic nervous system is under involuntary control. It is divided into two different sections: the sympathetic nerves, also known as the adrenergic nerves, and the parasympathetic nerves, also referred to as the cholinergic nerves. Our fat cells do not have cholinergic receptors. Our parasympathetic nerves release acetylcholine. Our fat cells do not respond to cholinergic receptors. Our adrenergic receptors are found on fat cells, and they are sensitive to the neurotransmitter norepinephrine. There are 5 subtypes of adrenergic receptors. They include 3 beta adrenergic receptors, beta 1, beta 2, and beta 3, and 2 alpha adrenergic receptors, A1 and A2. The 3 beta adrenergic receptors stimulate lipolysis, or the breakdown of fat, mostly intra-abdominal fat, than gluteofemoral fat. The 2 alpha adrenergic receptors stimulate, A1 and A2, stimulate lipogenesis, stimulate pre-adipocyte proliferation, and promote the storage of fat. More in the gluteofemoral fat section, or our lower body, than intra-abdominal fat. Your textbook says that this may explain in part why fat in the gluteofemoral area, or bottom half, pear-shaped people, it may be harder to lose weight around the hip area. The beta-3 adrenergic receptor is one of the 5 subtypes of adrenergic receptors. This particular receptor has thermic effects and anti-diabetic effects. We see mutations in beta-3 adrenergic receptors in those who are obese, those who are insulin resistant, and people who have type 2 diabetes. Lipoprotein lipase is an enzyme that is called the gatekeeper of the fat cells. Lipoprotein lipase hydrolyzes fats. It specifically splits apart glycerol from its fatty acids and promotes the storage of fatty acids. Levels will increase after eating, in the presence of insulin, and with exercise. Initially, levels will decrease after a fast or after dieting. However, if a prolonged low-calorie diet is followed lipoprotein for too long a period of time, for weeks, months, lipoprotein lipase will start to increase and promote fat storage. Lipoprotein lipase activity will vary among different fat deposits. There's higher activity in gluteofemoral deposits, especially in females, which can promote greater fat cell size in the hips and thigh area. Acylation stimulating protein is a small protein that is found in our plasma. It is responsible for promoting the storage of triglycerides. This protein actually rises in direct proportion to the body mass index, and it is found in higher levels in those who are obese. The levels of acylation stimulating protein will increase after a meal in response to an increase in blood triglyceride levels, and will decrease when blood triglyceride levels decrease. We see higher levels of acylation stimulating protein in those who are obese, those who are insulin resistant, and those who have cardiovascular disease. The factors that increase or decrease fat cell size are summarized here in this chart from your textbook. Lipoprotein lipase promotes triglyceride storage in cells, acts as a gatekeeper. Acylation stimulating protein also promotes triglyceride storage. It increases in obese individuals. And some alpha adrenergic receptors are anti-lipolytic and predominate in hip and thigh fat or gluteofemoral fat. Some beta adrenergic receptors work to decrease fat size and may have thermogenic and anti-diabetic actions. Leptin is an important protein in the regulation of body weight. It is produced by the fat cells. It comes from the Greek word leptos, which means thin. Lean people tend to have normal amounts of leptin in the blood, and they have hypothalamic receptors that are sensitive to this leptin. Leptin is found to be missing in mice that have been made genetically obese. Leptin is responsible for increasing the activity of anorexigenic effectors, or effectors that increase fullness and decrease eating. Leptin communicates to the brain how large fat stores are. And it's been found that obese people tend to have less responsive leptin receptors. This chart shows the negative feedback loop of leptin. We consume food. As a result or response, we increase the amount of leptin that's being produced. It travels to the hypothalamus. Peptides that are anorexigenic respond and increase POMC, alpha melanin stimulating hormone, and decrease NPY and agouti related protein. As a result of these peptides, we slow down our eating. Our leptin secretion drops, and orexigenic peptides respond, which act to increase appetite. And herein lies our loop. In some cases of obesity, too little leptin is produced. However, most people have expected amounts of leptin, most obese people. However, there is some disruption in how the body responds to it. For example, there may be impaired transport of leptin across the blood-brain barrier. Leptin receptors in the brain may be absent or severely reduced. Or we may have hypothalamic receptors that are insensitive to the action of leptin. When leptin levels are low, be reminded that it will stimulate food-seeking behaviors when they're low or when it's not being used properly. There are small proteins known as cytokines that can promote inflammation. Cytokines are secreted by adipocytes. Some of these cytokines are associated with increases in inflammation, such as tumor necrosis factor alpha, interleukin-6, resistant, and leptin. In those who are metabolically unhealthy and obese, they tend to produce more cytokines. Type of cytokine that is produced are called adipokines, cytokines that are produced by adipocytes. There are some adipokines that are actually associated with a reduction in inflammation. They include adiponectin. We see reduced levels in those who are metabolically unhealthy and obese. This chart from your book lists different adipokines that are released from adipose tissue and their effect and the levels that are found in the metabolically unhealthy obese population. The adipokines include the leptin, adiponectin, resistin, and tumor necrosis factor alpha. So we see disturbances in these adipokines in the metabolically unhealthy obese. The next topic in your textbook is body fat distribution and the influences of body fat distribution. Whether you store fat centrally or peripherally in your hips, thighs, and lower body is determined in part by gender and genetics. Central obesity or centrally distributed fat is more likely to change with exercise and lifestyle changes, while peripheral fat, that around the hips and thighs, is more resistant to change. This is a really great summary from your textbook that shows the differences in upper and lower body fat distribution patterns. Lower body fat distribution is also known as gluteofemoral or pear-shaped fat. Also, gynoid or peripheral fat are terms that are used. It's primarily seen in women. The fat cell characteristics usually include hyperplasia, growth of more fat cells. There are fewer beta-adrenergic receptors in body fat, or that is distribute in the lower body, or gluteofemoral fat. It is sensitive to the hormone estrogen. Estrogen can enhance fat storage. There is higher activity of the enzyme lipoprotein lipase, which promotes fat storage. There's less change evident with weight loss, and it's not associated with health complications until large amounts are accumulated. With upper body fat distribution, it is also known as internal abdominal fat or visceral fat, intra-abdominal fat, or android fat. It is primarily seen in males. The fat cells are typically hypertrophied cells. There is greater sympathetic nerve innervation when compared to lower body fat distribution patterns. Androgens like testosterone enhance fat storage, and fat cells that are distributed around the android region tend to be sensitive to cortisol, which is a stress hormone. There's greater change seen after weight loss with the upper body fat distribution pattern. It is associated with cardiovascular disease and hyperinsulinemia. Brown adipose tissue produces heat. It is a special type of tissue whose genes are regulated by a protein called PRDM16. This protein can increase the size of brown adipose tissue in humans. In laboratory studies where mice were exposed to cold, there was a proliferation of brown fat cells within storage fat. Brown adipose tissue promotes energy expenditure, so the more you have, the more energy you're expending. Defective brown adipose tissue may be responsible for contributing to some weight gain in humans. In laboratory mice that are obese, brown adipose tissue does not proliferate and is unresponsive. So there may be a defect there in humans. There are some genetic components that influence obesity in humans. There are about 11 single gene mutations that have been linked to a small number of obesity cases. There is a rare syndrome called Prader-Willi syndrome, and it is the most common of the genetically linked obesities. There's a defect in a segment of chromosome 15. People who have Prader-Willi syndrome have a seemingly uncontrollable appetite. Obesity is a polygenic trait, meaning that there are a number of genes that can work together to contribute to obesity. Some facial features common in Prader-Willi syndrome may include an eye abnormality called strabismus, almond-shaped eyes, a narrow temple distance and nasal bridge, thin upper lip, and downturned mouth. This is a review of the inheritance of genetic traits. Genotype refers to the actual genes that are available in an organism, while the phenotype refers to the expression of those genes within an environment. Excess production of certain products of our adipocytes may be inherited and influence body weight and body distribution. One example is lipoprotein lipase. Its production is inherited, and how we respond to this protein or enzyme when we exercise is genetically influenced. Lipoprotein lipase is an adipokine. Tumor necrosis factor alpha is another adipokine. Enlarged fat cells may cause an overproduction of it in obesity. This protein can promote inflammation, hyperinsulinemia, and insulin resistance. The fat cell receptors that regulate the differentiation of pre-adipocytes are also under genetic control. Our genes and our heredity favors weight gain for the survival of our species. A thrifty gene would be an advantage in times of food scarcity. Epidemiological studies support genetic components to obesity. And we have found that obesity-promoting genes act on the brain and its anorexigenic and orexigenic peptides. Such as leptin and POMC, etc. Some individuals may have low energy expenditures that has a genetic influence. Some people may have the tendency to conserve calories early in life. There may be some in utero factors that play a role. Physical activity is said to be 46 to 57% heritable. There are also some genetic influences to the effectiveness of various diets and food choice. There is another application in this chapter. If you're working with the 3rd edition, it's chapter 5. If you're in the 2nd edition, it's chapter 4. Part 2 is optional, just like part 1, but is a very good practice in drawing all of the information from the chapter together. So I encourage you to do the case studies. I may ask for you to turn in a case study as a bonus. So go ahead and take advantage of the optional case studies in the textbook. A good example of how environment and heredity work together includes the example of the Pima Native Americans. They are a similar group of people, one group living in Mexico with an active traditional way of life, and one group living in Arizona with a more Americanized lifestyle and diet. What we find is that the Pima Indians, Pima Native Americans who are living a more Americanized lifestyle with low fiber diets, low physical activity levels, and high saturated fat intake tend to have higher rates of obesity and chronic diseases, where we don't see that in the more active group of Pima Native Americans. Arizona Pima Indians or Native Americans have the highest rates of obesity and diabetes in the world. Also, Pacific Islanders don't do that well with Americanized diets. 75% of Pacific Islanders are diabetic. If you have a genetic susceptibility and to obesity and chronic disease, and you're consuming a high-fat, low-fiber diet, and your lifestyle is lacking in physical activity, then you are going to be more likely to be obese. Your prenatal environment may also influence your ability to store fat. In conclusion, and wrapping up this chapter on the regulation of eating behavior and body weight, know that there are biological forces that can override a person's willpower. In the most extreme case, Prader-Willi syndrome, individuals seem to have no control over eating. Some parents have had to padlock refrigerators and cabinets because their children with Prader-Willi syndrome had a very hard time controlling eating habits. There are differences between lean and obese people and receptors and effectors and regulating of genes. A modern lifestyle can overwhelm homeostatic regulation in a genetically susceptible person. We need to support obesity preventing activities such as a healthy diet and activity patterns, and the earlier the better. And finally, application 5.3, if you're working in the 3rd edition, or 4.3 if you're working in the 2nd edition, includes a case study on a guy named Joey who has inherited a defective gene and has been referred to a dietitian. Make sure you work through the objectives that are posted in Moodle before you attempt the quiz activity, and read and study this chapter in its entirety. Let me know if you have questions.